Good day, everyone. My name is Joy Concepcion from QPS LLC, and I will be the moderator for today's webinar. On behalf of QPS, I'd like to welcome you to Oligonucleotide Drug Analysis using Hybridization ELISA. This webinar will discuss the analysis of oligonucleotides by hybridization ELISA assays and how it differs from traditional ELISA immunoassays. It will be presented by Dr. Sandra Henkelman. A few housekeeping comments before we begin. Everyone will be muted during the presentation. Please enter any questions under the control panel. If we cannot get to your question, we will respond to you directly after this webinar. This webinar will be available on demand to all registrants. We will send you an email once the recording is available. And now I would like to introduce our presenter. In 2011, Sandra Henkelman obtained her PhD degree in biomedical engineering at the University of Groningen in the Netherlands. After a 4-year postdoc at the European Research Institute for the Biology of Aging, she joined QPS Netherlands in 2016. She is currently a team lead in translational medicine and supports the method development, validation, and sample analysis in the Department of Ligand Binding Assays and FACS assays. And with that, I would like to welcome Sandra.
Well, thank you, Joy, for the introduction, and welcome everybody to our webinar about oligonucleotide drug analysis using hybridization ELISA. So with this webinar, we want to give you some more insight in the analysis of oligonucleotides by hybridization ELISA assay. We show you how to design the capture and detection oligonucleotide probes, and we'll give you more insight into development and qualification of methods for the analysis of oligonucleotides in various tissues. So for the last 2 to 3 decades, oligonucleotides have been utilized for their therapeutic properties, and every year more studies are emerging. The oligonucleotide drugs are used to treat a wide variety of diseases, including cancer, but also inherited diseases or cardiovascular diseases or muscle diseases. So oligonucleotides are an emerging therapy. So what are oligonucleotides? Well, they consist of a short single or double-stranded sequence or oligomers of synthetic RNA or DNA. They usually are up to 15 to 30 nucleotides long, but they can be longer as well. But they rarely don't exceed 200 nucleotides. There are various oligonucleotide therapeutic approaches available to quantify the oligonucleotides. One of them is the antisense oligonucleotides approach, also referred to as ASO. We also have small interfering RNAs, referred to as siRNA. Also microRNAs are used as an approach. Splice switching oligonucleotides, aptamers, and GCP desoxynucleotides, but also more recently CRISPR-Cas9 approach. So there are a lot of different approaches, and the primary mode of action of these oligonucleotide drugs include gene silencing, splice modulation, or gene activation. So up to date, there have been 10 oligonucleotide therapeutics being approved by the FDA. And what you can see is that different approaches have been used for these treatments. So, for example, you have a first-generation ASO, but also spherical block ASO. You see gapmers ASO, but you also see aptamers being used, or small interference RNAs. The size of the oligonucleotides that are being used varies from 18 to 30 oligonucleotides, so they're not really big. And as mentioned just before, they're used to treat a wide variety of diseases. So the first one that was approved was actually for the treatment of the inflammation of the retina, but you see also that treatments have been used for some cardiovascular disease, muscle disease, some inherited disorders like TTR, where you produce abnormal protein called amyloid and being built up in your tissues and organs. So overall, a variety of diseases that are being treated with these oligonucleotides. And what is interesting to see that for some of the same diseases, different approaches have been used. So, for example, for the TTR, you see that sRNA approach is being used, but also the gapmer Assays. So, and with the spinal Duchenne muscular dystrophy, you also see two different oligonucleotides that have been approved. One is focusing on exon 51, the other one is focusing on exon 53, but both they use a similar approach, namely the spherical block ASO. So with this in mind, recently there was a survey by Bioanalysis Zone, and from that you can see that the most widely used oligonucleotides approaches are the ASO with 59%, but also the small interfering RNAs. What you also see is that compared to 2019, less oligonucleotide studies have been performed in their portfolio, but this could also be dependent on the type of response they included. So what we do see is that more recently there is a focus on the rare genetic disorders and the cancer inflammatory disorders, so a bit of an increase, but also still treatments are available for cardiovascular and infectious disease. So the most widely used therapeutic oligonucleotides are ASO and small interfering RNAs. Below is a picture, a schematic diagram of how what the mode of action is. So with the left picture, you see a single-strand oligonucleotide that is being hybridized to a complementary sequence via the Watson and Crick base pairing. Once it is being hybridized, it can either block the ribosomes or other factors, which is called spherical block. Also, protein factors can be recruited, like RNase H, or it can either modulate the splicing process. Well, with the right picture, you have the small interfering RNA, you have a duplex RNA, which is being associated with AGO, an RNA endonuclease. In this complex, the passenger strand is removed, and the guide strand is leaded to the target, where it binds with a complementary sequence. Well, if it binds perfectly, then the messenger RNA is cleaved and degraded. If it partially mismatched, then you have associated mediated repression. All in all, this is a very powerful tool to silence your gene. So I want to tell you something about oligonucleotide quantification. So there are different bioanalytical methods available to quantify oligonucleotides. This is, for example, the LCMS method, the liquid chromatography, mass spectrometry, but also the HPLC with fluorescence detection, or the qPCR, but also the hybridization ELISA. So traditionally, hybridization of ELISA is being used to analyze the large molecules from bioanalytical matrices, but an LCMS for the small molecules. However, recent advances in instrumentation, but also sample preparation techniques, has propelled LCMS as a complementary method and is being used for the bioanalysis of oligonucleotides as well. Well, with this in mind that there are different platforms, techniques to measure your oligonucleotides, you need to think upfront and evaluate case by case which platform you need. This mainly depends on your scientific question. So, for example, which sensitivity do you want to reach? But also, for example, for the resource availability. So, which technique do you have in house? Do you have experience with it? Are the reagents easily obtained? And what do they cost? So these are all questions which are mutually important. And last but not least, it's also important to understand the strengths and your limitations of your techniques. So where one technique can be the choice for one client, can be that the other technique is more needed for another client. So keep this, please keep this in mind. Well, with this presentation, we focus on the hybridization ELISA, and I will tell you something more about that. So the hybridization ELISA, that's a type of ligand binding assay, and below you see a picture of how it's in practice. So you have a diluted sample, which is being immobilized using a capture reagent. Unbound reagent is washed away and labeled detection reagent is added. In the end, a complex is formed, and this complex is, after an extra wash, detected by addition of substrate. So when you add the substrate, a signal develops, which could either be a color, a fluorescence or luminescence, or a light signal, which can be detected with an ELISA reader or an MSD ECL electrochemiluminescence reader. So the hybridization ELISA is the preferred method for the quantification of therapeutic oligonucleotides. This is mainly due to the excellent sensitivity in the picogram per mil order and the high sample throughput. With this setup, the antibodies in ELISA are replaced with labeled complementary capture and detection oligonucleotide probes. There are various hybridization ELISA assay formats to quantify oligonucleotides. You have the 1 and the 2-step hybridization assay. You have a sandwich hybridization assay, a dual ligation, or a competitive hybridization assay. In the next 2 slides, I want to explain a bit more about the 1 and the 2-step hybridization assay and the dual ligation hybridization assay. So with the 1-step hybridization ELISA, what you have a complementary probe with at one end a biotin label and the other end a detection label, which is being hybridized against the analyte oligonucleotide. As a nuclease is added to cleave away unbound oligonucleotides. So with this technique, you cannot differentiate between the intact oligo measured in vivo and 3% truncated metabolites can form. So you will get an overestimation of your intact oligonucleotide. With the 2-step hybridization ELISA, you start with a capture probe, which is biotin-related at one end. This hybridizes against the analyte oligonucleotide. And again, the whole complex is being immobilized on a streptozidine-coated plate. Afterwards, the detection oligoprobe is added, and which binds to the complementary 5% overhang to the analyte oligonucleotide. And the whole non-ligated oligonucleotides are washed away. With this method, you cannot distinguish between the intact oligo and the 5% truncated metabolites. So also here you will get an overestimation of the intact oligonucleotide. With the dual ligation hybridization ELISA, you start with a template probe with a 3 and a 5% overhang, which hybridizes against a biotin-related capture probe and to the analyte oligonucleotide. The whole complex is immobilized. on the streptococcus plate, and the detection oligo probe is being ligased against the analyte oligo. With this method, there's no interference from the 3 or the 5-oxent metabolite, because only the intact oligo can be is quantified. So over the years, various modifications and improvements have been made in the assay formats to improve the assay sensitivity, but also to decrease interference. So in our lab, we use a modified 2-step hybridization procedure using the MSD-ACL platform. The nice thing about this is that it increases your dynamic range, which allow for less further dilution. So below is a schematic picture of it. So what we start with is a biotinnylated probe, capture probe, which is being immobilized on a streptococcus plate and hybridizes against the analyte oligo. So unbound oligos are washed away, and the DIG label detection probe is being added and is also hybridized against the analyte oligo. After the wash, to remove unbound oligos, the whole complex is detected by a Silvertech labeled anti-DIG conjugate. So that means that if a voltage is applied, the bound Silvertech will emit light, and the concentration of emitted light is being measured. So next, I want to tell you something about probe design. So with our 2-step hybridization ELISA, you need to develop a separate capture and a separate detection probe. What is important, that the sequence is complementary to the analyte, that half of the length of the analyte is being used to create that, to design that probe, but also that you include a biotin label and to be able to use it on streptococcus binding plates. It's also good to include a TEG spacer and tetraethylene glycol spacer in order to improve the accessibility. So what it does is to create a big distance between your biotin label and your oligonucleotide in order to be able to better bind to your streptococcus plate, but also better to your analyte oligonucleotide. With the design of the detection probes, it's kind of similar. So the sequence should be complementary to the analyte. Also, half the length of the analyte should be used, not complete. And with the detection probe, a DIG label needs to be added, so you can use anti-DIG reagent to generate an assay signal. When you set up your, when you design your probe, it's handy to create 3 different probes of each, so 3 different capture and 3 different detection probes, and then you evaluate for optimal binding. So when we talk about probe design, it's important to keep in mind that probes should be optimized for binding and stability. So you should prepare a fully phosphorylated probe. This increases the stability of the probe. Every probe should include 5 LNA bases. LNA is a locked nucleic acid base, and what happens is that a linker is being formed, which locks the conformation of the molecule and therefore increases the binding of the oligo. You could also include methyl bases for non-LNA bases. That means that you can either have LNA bases or methyl bases. You cannot have them both. And you see that in this picture, where a methyl group is added to 2-hydroxy of the ribose ring. So by doing this, the probes becomes more stable because the melting temperature is being raised. If you design your probes, it's also important to estimate your TM. U2M is your melting temperature. In general, the higher your melting temperature, the better the binding of the probe, so the more stable it is. There are different formulas to calculate this. If you, for example, have a small oligonucleotide, so 13 or nucleotides or smaller, you have a formula where the amount of A plus the amount of T in your oligos multiplied by 2, plus the amount of G, plus the amount of C in your probe multiplied by 4. So you can calculate your melting temperature. If you have longer oligonucleotides, the formula is a bit more complicated, but you can see that if you have a lot of GCs, for example, in your probe, that your melting temperature is a bit higher, which increases your stability. Well, I will show you an example in the next slide, so to guide you through it. So if you have a sequence of the analyte, you write that down. What you do is you write the complementary sequence. So the complementary sequence is right here. Then you rewrite it the other way. So it's just a different manner of how you presented it. And then you start with designing the capture probe. Well, we talked about including a biotin label and a text spacer for the capture probe. So let's start with designing the probe. So if you have, this is your sequence, then only one thing you should also take into consideration is that the T is not used in the probes, or the T is used in probes because the uLNA does not exist. So if you have a sequence like this, that means that your capture probe will be G, T, A, T, C, T, T. So you make sure that all the nucleotides are phosphorothioated. That's important, so the capital. But you also want to include the LNA bases, as we just discussed. So we include 5 LNA bases, and they can be phosphothioated bases, but also LNA bases. But as mentioned before, if you add a methyl group, this is one way or the other. So you cannot have both. So we added here methyl groups as well, and this is our first capture probe being designed. If you do that for the detection probe, it works the same. So you have your sequence, which would be ACC. This becomes a T, ATCC. So that's written down here. These bases should all be phosphothioated. Again, we add some LNA bases, which are depicted in blue. And we want to have some bases with methyl groups, which you put in red. So this is actually how you can design your capture and detection probe. It's also good to keep in mind that if you design them and you order them, it's good to check with your company who synthesized this probe what the melting temperature is, and what if they have additional information for you. Okay. So next, I want to tell something about qualification of oligonucleotide assays. So assessing the biodistribution of oligonucleotides is a crucial part of any preclinical development program. If you want to quantify the oligonucleotides in various matrices, you can think of serum and plasma, of course, but also kidney and liver, where the oligonucleotides may accumulate, but also tissues of interest. And what I mean by that is if you have an oligonucleotide drug for muscle disease, then you also need to quantify your oligonucleotides in muscle tissue. If you have a rare eye disease, you also need to quantify the oligos in the different eye tissues. So you need to keep that in mind. So while there is minimal sample preparation for the quantification of oligonucleotides in serum and plasma, there are sample preparation steps needed for the quantification of oligonucleotides in tissues. This can be done by liquid-liquid extraction or, for example, protein K digestion, where the proteins protein interference is avoided, which allows for a better binding. So what we do in our lab is actually we prepare tissue samples for the quantification of oligonucleotides via proteinase K digestion. So what we do is we prepare homogenate and we combine that with the proteinase K. So what we do in short is we weigh a certain amount of tissue. The tissue is being added into a Parcellus tube, which contains beads. These beads are available for a variety of tissues. So if you have a rigid tissue, you need different beads than if you have a tissue which is more soft. So you can check that up front. So the tissue is in the tube. Extraction buffer is added, and the homogenization steps is ongoing. And after this homogenization step, proteinase K is added, and the amount of proteinase K that is added is dependent on the weight of your tissue. So once the proteinase K incubation has been performed, the sample is inactivated, for example, on a heat block, cooled and centrifuged so that the obtained tissue homogenate is a clear fluid. The way we design the oligonucleotides is in such a way that they are resistant to DNAs and RNAs, allowing extraction from tissue without causing any oligonucleotide instability. So, regarding the guidelines for the qualification of oligonucleotide assays, there are two major guidelines that are available. Is the bioanalytical method validation guidance for industry from the FDA, but also the EMA guideline on the bioanalytical method validation. If you need to qualify your oligonucleotide assay in serum and plasma, these guidelines are really helpful to validate your assay. If you want to qualify the oligonucleotides in tissue samples, then a fit-for-purpose approach is the way forward. While the EMA doesn't talk about the fit-for-purpose, the FDA has listed that the fit-for-purpose concept states that the level of validation should be appropriate for the intended purpose of the study. So that's not much to work with. So what we did is we want to guide you a bit through two case studies of us, which we set up oligonucleotides in tissue samples, and to give you an idea what is needed. So in the first case study, an oligonucleotide assay was set up for an ocular disease. A client developed an RNA oligonucleotide to potentially treat a genetic ocular disease. The mode of action is that the RNA oligonucleotide modulates the RNA splicing process, thereby correcting the gene defect and produces a functional protein. We used the antisense oligonucleotide method for this, and we designed a two-step hybridization ELISA assay in various monkey eye tissues on our ECL platform. So for the fit-for-purpose approach in tissue homogenate, it's not so different than a validation. It's just a bit less extensive. So what we did, we made a calibration curve with a 4PL fit and a weighing factor of 1 divided by I squared. We tested for precision and accuracy at 5 different levels. So the intra-precision was measured 5 aliquots of each level in 1 run, while the inter-precision was measured for 2 aliquots of each level in 5 runs. So normally we would do in a validation 6 runs. Selectivity and recovery was measured at two levels, LQ and high, and we included 3 individual homogeneous samples of each level. This is also a bit different than a validation where most of the time 10 matrices Blanco matrices are used and be tested. We also tested dilution linearity. We spiked a sample at ELISA and tested 5 aliquots at different dilution. And we tested short-term stability. So when you talk about short-term stability, this actually includes the benchtop stability. So we tested 3 aliquots for the benchtop stability for 24 hours, and we tested freestall stability up to 3 freestall cycles. So these are a couple of results we had. The fit-for-purpose approach is the Hoyt tissue homogenate as an example shown. So the precision and accuracy was measured at 5 different levels, from LQ to ULQ, and low, mid, and high. For the precision and accuracy, the acceptance criteria states that the intra and interim precision and accuracy should be less than 25%, and the total error, so the sum of the diffie and bias, should not exceed 40%. Well, you do see that both for the intra-run and the inter-run, the acceptance criteria are met, and that's actually just for all levels. So this means that the assay shows a good samples show good precision and accuracy in this setup. If you look at the selectivity, which was measured at LQ and high level, you see that the mean recoveries was also within the acceptance criteria, meaning that the assay is selective for the oligonucleotides. If you check the short-term stability, you see that at time point 0, the mean recoveries are nicely around 100%. You see that the benchtop, for the benchtop, the mean recovery is also accepted and within the 75 to 125%. So that means that you can store samples at room temperature for at least 24 hours. And you do see that the freeze thaw cycles show acceptable recovery, indicating that samples can at least be freeze thawed 3 times. If you go to the dilution linearity, we initially used the 20-fold as an MRD and a minimal prior dilution. You see that's nearly 100% as a mean recovery. And you do see that you can dilute up to 12,500 fold, because these are the mean recoveries that still fall within the acceptance criteria, but at 25,000 fold, it's outside the acceptance criteria. So the assay for the determination of oligonucleotide in a monkey whole tissue homogenate, you see that samples can be analyzed with good precision and accuracy. The assay is selective for the determination of oligonucleotides in whole tissue homogenate, and homogenized whole tissue samples can be diluted up to 12,500 fold. They can be frozen and thawed for at least 3 times, and they can be stored at room temperature for at least 24 hours without showing any deterioration. If we go to our second case study, we set up an oligonucleotide assay also for nuclear disease, but a slightly different one. And what we did is we used whole eye homogenate for the determination of oligonucleotides in various monkeys' eye tissues. We wanted to know if you could use the whole eye tissue homogenate as an alternative for all the individual homogenates of the individual eye tissue homogenates. So what we did is we set up again a 2-step hybridization assay in monkey whole eye homogenate using our ACL platform. We prepared calibration and QC samples from the whole eye homogenate, and we quantified oligonucleotides in various monkey eye tissue samples. So we started with the whole eye tissue homogenate and qualified this as well. We tested the calibration curve with the 4PL fit and a 1 divided by I-square weighing factor. We tested again intra and inter-precision and accuracy at 5 different levels. The selectivity and recovery was measured at 2 levels, and the dilution linearity was the sample was spiked at ULQ, and also the short-term stability was tested. So the benchtop and the freeze thaw stability. So once this was qualified, we wanted to verify in individual eye tissues if the assay showed good precision and accuracy. So we tested inter-precision and accuracy, but also that the assay was selective enough. So we tested selectivity and recovery at 2 different concentrations. If you look at the precision and accuracy result of the whole eye tissue homogenate, you see that for the acceptance criteria, for all concentration levels, the CV and bias is below the 25%. And you see that the total error of CV and bias together is below 40%. So indicating that there's good precision and accuracy with this whole eye tissue homogeneous assay. If you look at selectivity, this is the same picture. You have you measure two different concentration levels, but you see that the mean recovery falls within the acceptance criteria of 75 to 125%, indicating that the assay is also selective for the oligonucleotide. We have a lot of data with also comparable results. I'm just going to show you the retina choroid tissue, but we also tested a lot of other eye tissues, but they give similar results. So I'm going to show you those two. And the precision and accuracy for the retina choroids also are all within the acceptance criteria. So CV and bias are below 25% for all concentration levels. And the total error is below 40%. You do see a bit more variation with the choroid tissue, having a bit higher CV than in the retina, but again, it's all within acceptance criteria. So the precision and accuracy is for those tissues is good. If you track the selectivity, and we show here the selectivity in two different concentrations, LQ and high, both in retina and in lens tissue, you see actually that the mean recovery is also very nice and falls within the acceptance criteria, meaning that the assay is also very selective for individual eye tissue homogeneity tested. So with the verification of this whole eye tissue homogeneity assay and individual monkey eye tissue homogeneity, you see that the assay shows good intra-run precision and accuracy for the individual monkey eye tissue homogeneity samples tested. And the assay is also selective for the determination of oligonucleotides in individual monkey eye tissue homogeneity samples. And what does it tell us? This means that this allows us to quantify oligonucleotides from various eye tissue homogeneity using the whole eye tissue homogeneity assay. And this is handy because if you have to homogenize all the individual tissues and qualify them, that's a lot of work if you have a lot of different eye tissues. And if you can use the whole eye homogeneity assay, then yeah, this is of course less work to do. So in the last two slides, I want to tell you something about challenges in the biotransformation assessments. So what are the challenges? We talked a bit about the formation of metabolites. So oligonucleotides can undergo cleavage of phosphodiester bonds, leading to shorter, also called truncated metabolites. So to tackle this problem, you can perform cross-reactivity and interference testing of metabolites. So with cross-reactivity, what you do, you add the metabolites to your assay. You don't include the intact oligo, and you check if the metabolites are being picked up in your assay. With the interference test, you have the metabolites, but also the intact oligo in there, and it will show you if there's any interference from the metabolites in the assay. So when you set up these assays, what you do see is that metabolites are being formed up front. So you have your full intact oligo, you have an N-1, you have an N-2 from the 3' end, but also from the 5' end. And you can do this up to 5, 6 oligonucleotides being removed. And you can test those in the cross-reactivity and interference. What you can also do is use of a different hybridization assay setup, for example, the dual ligation hybridization assay setup. And with this setup, there's supposed to be no interference from either 3 or 5% truncated metabolites, because only the intact oligo is being quantified. You could also use different platforms or in combination with the platform. And use the LCMS platform to identify and characterize truncated metabolites. So it's not sometimes the one way or the other. You can also use different platforms to answer your scientific question. Well, for the assessment of oligonucleotides in tissue sample, no long-term stability is obtained. And this is because the stability of the tissue homogeneity is not really representative of the stability of the tissue. During the homogenization process, enzymes are released which could affect the stability of the homogenate and which does not nearly reflect the stability of the tissue. So that's one point, but also due to the homogenation step, you lose the possibility to evaluate regional distribution. So most of the time you have a tissue and you cut a piece of the tissue and you homogenize that. And the concentration which is measured is, of course, one part of that whole tissue, because it doesn't really reflect what's in the whole tissue. With this in mind, no long-term stability is obtained. However, if you want to measure your homogenate, your tissue homogenate on a later time point, it is wise to probably do some long-term stability testing to include that. Although there are no clear guidelines for determining the stability of tissue homogenates, this is an ongoing debate in the bioanalytical community and point of attention. So we will see more in the coming future. So I want to close down with my last slide and tell you that for the appropriate bioanalytical strategy must be implemented for the quantification of oligonucleotides. That means that sometimes you need different platforms to quantify your oligonucleotides because there are so many different out there, and they can there are different types, depend on their size, chemistry, and complexity. So keep in mind that you can use different platforms to answer the questions. And with it, I want to thank you for your attention. And I hope I gave you some more insight in the oligonucleotide drug analysis using hybridization ELISA.
Thank you, Sandra. And now we will begin the question and answer portion of the webinar. Please enter any questions under the control panel. Our first question: how do you recommend which hybridization approach is selected?
Well, that depends on what your starting point is. So sometimes if your client already has done some work, you know you start off from there. If you have to start from scratch, then one of the things to keep in mind is do you have experience with the technique? Or, for example, we have a lot of experience now with the two-step hybridization ELISA. Yeah, for us that works. So you keep on going there and moving forward. So it depends mainly on what is your starting point.
Okay. And do you centrifugate the tissue samples before running samples by hybridization ELISA?
Yeah, so what we normally do to obtain the tissue homogenate is that the last step, we have a centrifuge step. So to obtain the homogenate, we have a centrifuge step. So normally we don't need to centrifuge the samples when we have them in the assay. We had one incident where it was needed. We saw a bit of tiny debris, and there the centrifugation really led to better results. But it's not standardly being implemented, but it can be. Yeah.
Got you. And could you please elaborate on how the ocular tissues are homogenized?
So, for example, we have set up different assays, and you get, for example, the retina tissue, or you get the lens, and you need to. What you first do is you weigh a certain amount of tissue, and you put that to the Procellus tube for the homogenization step. So with the beads in there, they will squeeze and wash the material, and that helps to homogenize the sample. Now it depends on how rigid your sample is. If you have samples, tissue samples that are more rigid, then you different Procellus tube with different beads in there to get the tissue all well, degraded and homogenized.
And would you happen to know what the dilution factor was for the I-homogenate that you had tested?
So for the whole I-homogenate, I think it was 20 from the top of my mind. For the individual eye tissue samples, it ranges from 10 to 20. So yeah, it's all in the same range.
Okay. And the melting point calculation does not count modifications like LNA, PS. And so how is the melting point estimated?
So you, of course, you can make self calculation for yourself, but when you design the probes, it's also good to keep in contact with your probe oligonucleotide synthesizer. So the company that makes your probe, what we normally do is we show them upfront which probes we designed, and they come with also a good estimated melting temperature. So they keep that, they're able to implement that in their formula, I think, and they share that back with us. So upfront we know which probes are having which melting temperatures, and if we see, for example, that one probe has a really low melting temperature compared to the other two, we would rather design another one instead of using the lower melting temperature probe.
And if the LLOQ is 1 nanogram per mil in tissue homogenate, how does it translate to the amount in the original tissue?
So tissue is being homogenized in a certain amount of buffer. So for example, if we have 1 microgram of tissue, we add 100 microliters of buffer. This will give you a 10 microgram per mil concentration, and with this concentration, you can count back how much microgram per grams oligonucleotides should be in your tissue. So you measure the nanogram per mil, but because you have a steady concentration being homogenized, you can use that to calculate back to micrograms per grams in the tissue.
Got you. And how long does method development typically take for hybridization ELISAs in comparison to traditional ELISAs?
So it doesn't take too much longer. So with a traditional ELISA, you most of the time have your all your reagents in house. With the oligo method development, you need a few extra days to test the different combinations of oligonucleotides to get the optimal binding. So regarding that, it's only a couple of days.
And where do people typically get their oligos, and how much time does the probe design and procurement add to the overall method development timeline?
So there are a lot of good companies out there that can synthesize the oligonucleotides. We have good experience with Eurogentech. What I like about those guys is that if you have a question or you're not quite sure if your probe design is the best way forward, they're very patient with you and help you with that. And I think that should be key if you find a company that synthesizes your probe that really helps you think with you instead of not helping you. So yeah, so we have good experience with that. And how long does it take? So normally, if we design the probes and we have to order them, it takes 2 to 3 weeks before we receive them. Again, you have to test the individual combinations. And then if you know the right combination, you order a big bulk of that. So then it will take another 3 weeks before you receive them. So it's not much more days you need in the end, but the timeline is a bit more longer because you need to order the oligos.
So essentially you're ordering them twice. First,
Yes.
mini batch, and then once you select which ones you're going to use, you order a larger batch.
Yeah, because you don't know upfront what is the right design of probes, and you first need to test that before you order a big bulk.
And one last question, how long do you incubate with proteinase K?
From the top of my head, I think it's around 45 minutes. Could be around an hour, but I need to check that. But yeah, so in that order.
Okay. All right. I'd like to thank everyone for their attendance today. Like I said, we will have this recorded and let everyone know when it is available for on-demand viewing. And with that, I would like to wish everyone a great day.
Thank you, everybody, for joining.